There is a term in computer science for badly written code. Code that works, but that no one really knows how it works anymore. Inherited code accumulated over decades, made of pieces that intertwine, overlap, and depend on each other in ways that no one has ever documented. It is called spaghetti code. And according to Adrian Woolfson, molecular biologist and author of a book published last week by MIT Press, the human genome, the code of life itself, is exactly that. A disaster accumulated over 4 billion years. But for the first time in history, we have the tools to rewrite it. To understand why the genome is a disaster, you have to understand how it was written. Evolution does not design. Evolution does not refactor. Evolution keeps what works and adds more things on top of it, endlessly, for 4 billion years. The result, Woolfson says, is that if an engineer looked at the human genome for the first time, he would say, and I quote literally, "My God, what an absolute mess." Components that do several things at the same time, functions hidden inside other functions. Sequences that seem useless, but if you remove them, make everything collapse. Think of a piece of software opened for the first time after 4 billion years. No comments, no documentation, and you cannot shut it down because it is you. The first to seriously try was Jeff Boca, an American biologist now at New York University. 15 years of work, an international consortium involving laboratories in 5 countries. The goal? To rewrite from scratch in an orderly way the entire genome of baker's yeast, not modify it. Rewrite it. 16 chromosomes, 6,000 genes, 12 million bases. Clean, organized, readable. The project is called SC 2.0, and it was completed at the beginning of 2025. But Boca did not stop at copying. He added, natural yeast has 16 chromosomes. He built a 17th one, completely new, which does not exist in nature. And in a separate experiment, he showed that the 16 chromosomes can be taken and fused into just 2 gigantic ones. A total reconfiguration of how genetic material is organized. It works. The yeast lives. What has changed in the last 2 years has a name: genome language models. They are artificial intelligence models that work like ChatGPT, but instead of working on the 26 letters of our alphabet, they work on the 4 letters of DNA. A, T, C, G. The most powerful was published in Nature in March 2026 by the ARC Institute. It is called Evo2, 40 billion parameters, trained on 9 trillion bases of DNA from all known life: bacteria, plants, fungi, animals. But the number that really matters is another one, the context window, 1 million bases. To understand why this matters, you need to know something that DNA does and human language does not. In a book, the meaning of a sentence is found inside the sentence. In DNA, one sequence can influence another that is located 1 million letters away. It is a conversation at a distance, and to read it, you need an AI that can keep everything in mind at the same time. Evo2 can. Woolfson uses a metaphor that is worth listening to in full. He says that the hardest problem today is not writing DNA. It is understanding the grammar of DNA, a grammar that no one has ever written in a manual because it formed by itself over 4 billion years across all living species. To learn it, we need enormous databases of sequences from as many forms of life as possible. And here is the point that leaves a bittersweet taste. I quote Woolfson, "Every time we destroy a species, we destroy a page of the grammar book. We must become grammar hunters." The conservation of biodiversity is not only an ethical or ecological issue. It is also the conservation of knowledge that, without those species, we may never be able to learn. Woolfson makes a prediction. Within 50 years, he says, biology will be the engineering material of choice. Not metal, not concrete, the living. He gives a concrete example. Spider silk already has, in nature, the same tensile strength as steel. Redesigned with artificial intelligence, it could become 5 times stronger. And with one advantage that steel will never have, it can be intelligent. It can respond to the environment. It can repair itself. Imagine steel capable of noticing when it is failing. Imagine buildings that heal themselves, clothes that change according to the weather, houses that, instead of being built, are grown. Some of these things are decades away. Others, already today, are in the first prototypes inside laboratories. Woolfson is also about two kinds of failure. The first is mechanical. When you clean up the code of life, you risk removing redundancies that were useful. Biological systems are built to be robust. They have overlapping rescue mechanisms. Removing them, even to make them more elegant, means making them more fragile. The second kind of failure is more serious. I quote Woolfson again, "We do not understand ecosystems. They are incredibly difficult to calculate. If we release engineered organisms into complex ecosystems, we can create disasters." And he adds that these technologies in the wrong hands are dangerous by definition. The promise is enormous, but it must be handled with a level of caution that so far the technology industry has never shown it knows how to have.